Great. Welcome to this month's March Mycotalks. We have two outstanding speakers today. On behalf of my co-chair, Dr. Judith Berman at Tel Aviv University. I'm Jay Coles at Tulane University, and the two speakers are Dr. Julie Segre from NIH, and she's going to talk on the human skin microbiome, finding friends and foes. And then Dr. John Perfect from Duke University, going to talk on the sugar-coated killer yeast unplugged. So many participants have probably been a member of the Mycotalks community for a number of years now, but the philosophy and origin is that we foster diversity, including diversity of career stage, gender, geographic diversity, and also, of course, diversity of the field of research. We have two 30-minute talks on all aspects of medical mycology, and then 30 minutes of in-depth discussion. Please submit your questions through the Q&A button, which will be moderated by both Dr. Berman and myself, and recordings will be available on the MRC CMM YouTube channel. Remember that for each month's Mycotalks, you do have to register for each session. And for in April 27th, we'll have Dr. Yinping Shui from Taiwan, and then Dr. Junquan Chung from the National Institute of Allergy Infectious Disease. And then on May 25th, we'll have Georgios Kamilos from Greece and Anurada Chaudhary from University of Delhi in India. And I would also like to mention that medical mycology trainee series. This is for students, postdocs, clinical trainees, and new faculty to present your work virtually to the global mycology community. These talks are held on the second Thursday of the month, but timing may be adjusted to fit speakers or time zone. And so help us highlight this helps us highlight the scientifically broad and international medical mycology community by, and please nominate speakers that you think would be excellent talks for this program. And again, we foster wide scientific interest in global diversity as part of this trainee seminar series. And you can get in touch or submit your abstract with the link highlighted here below. So with that, I would like to turn this over to my co-chair, Dr. Judith Berman, who's going to introduce our first speaker.
Thank you very much, Jay. And it's a pleasure to be here. Good morning, good afternoon, and good evening to all of you. I'm very pleased to be introducing Julie Segre, who is the senior investigator at the National Human Genome Research Institute at the NIH. Julie received her bachelor's in mathematics from Amherst College, her PhD in genetics and genomics from MIT, and did postdoctoral training in skin biology at the University of Chicago. She serves on the American Society for Microbiology board and is a member of the National Academy of Sciences and the National Academy of Medicine. Her research focuses on the human microbiome, investigating both the transmission of hospital pathogens and the diversity of commensal skin organisms, including bacteria, fungi, and viruses. Her research integrates DNA sequence technology, algorithm development, and clinical studies to explore the diversity of microbes in and on humans in health and disease, with her current research focusing on the emerging fungal pathogen Candida auris in health care ecosystems. I want to remind you again to put questions in the Q&A section below, and try and put the name of the speaker for whom they are addressed. We may combine some of the related questions, so you may not hear your question addressed individually, but the speaker will get all of the original questions after the session. So welcome, Julie, and please load up your talk, and we'll be happy to hear it.
Hi, good morning, good afternoon, good evening. Thank you all for joining. And thank you for that very nice introduction, Judith. And I see that participants can now see my screen. So I'll be talking today about the human skin microbiome and finding friends and foes. And great. So what is the human microbiome? When we talk about microbiome, and when I use that term, I'm talking about the collection of all the microorganisms and their DNA living in association with the human body. So I mean, for the skin, we find fungi, we find bacteria, we find viruses. In the human skin, we actually find appreciable amounts of fungi, and so we've really been trying to lead the way in characterizing those. I know for a lot of people, the human microbiome started by meaning basically the bacteria that live in your gut, but we've really been trying to lead the charge on thinking about the 3 different kingdoms of microbiota. So I want to just start by talking about how do we characterize the microbiota, because as Judith mentioned, my training is really in genomics and in skin biology. And so we really have been trying to iterate through the different ways in which we can characterize the microbiota. And that's going to be the focus of my talk today. So I'm going to talk about the 3 different ways of characterizing a microbiome and give you examples of those in our own research. So I'm going to talk about amplicon sequencing, shotgun metagenomic sequencing, and isolate sequencing. So I want to just start by saying that all 3 of those methods have the origin of the skin, which is depicted here in the middle. So maybe I should use the laser pointer. Okay. So the bacteria, fungi, and viruses that live on the surface of the skin, but they also live deeper into the skin inside hair follicles, sebaceous glands, and so on. And I'm going to start by talking about how we started, which is by sequencing the fungal ITS1 region. Okay. So just a brief survey of a brief sort of run through of how we do this. Because this is something that I just thought was at least something we really tried to create standards for. So we use foam swabs and lysis buffer, and we swab humans. And we do this all through clinical protocols here at the NIH Clinical Center, seeing healthy volunteers and working with other people who do this also through clinical studies. Swabbing the skin, and then that foam swab goes into lysis buffer. Genomic DNA is made directly from that swab. There's no culturing. And then in the case of the ITS1, which we have published, we use the ITS1 primers, and the ITS1 sits between, as you saw in the last slide, the ITS1 region sits between the 5.8S and the 18S gene in the ribosomal RNA genes. So very similar to how we're amplifying the 16S gene for bacteria. It's a signature gene. Every fungi has an ITS1 region, and but that's an important question, is like, how well does this work? And so I would say here on the right, I tell you about a mock community that we obtained that has equal molar amounts of 10 different fungi. And we always use this mock community. We have now ourselves also made a different mock community for skin-specific studies that has a number of fungal species of Malassezia and Candida. And I just highly encourage people to have a mock community so that you have a positive control and you know what you're amplifying. So each of the samples is amplified with ITS1, and the ITS1 region is labeled with a barcode. Again, we've have more details in some of our publications, but you basically have a staggered barcode on each primer that uniquely identifies that sample. We do our sequencing for these amplicons on the Illumina MySeq, and we use the barcodes then to assign the samples back. And then we're going to assign each of those reads to a species level and count the relative abundance. So we'll get a species by sample matrix. Okay. So doing this from the original studies of the human skin, we look at the skin as an ecosystem. And so we have multiple different body sites of the skin. And here with the bacteria, we showed that the bacterial communities were specific to the body site in terms of them being oily, moist, or dry. But on the for the fungi, what we really found was that it's pretty much Malassezia dominated on the core body sites, and the diversity of fungi that we're seeing is in the feet sites, the toe web, the toenail, and the plantar heel. I show that in a little bit more detail in the next slide. So these are published. I'm just going through it really quickly, but basically for the Malassezia, on the left of the body sites, you can see 10 different healthy volunteers, and it's this darker purple, which is Malassezia. And then on the right-hand side, it's those same samples, but now I'm showing you the species level so that you can see that we can identify Malassezia restricta, globosa, and see some specificity where you're seeing here in the external auditory canal, that's the Malassezia restricta, but here you're seeing Malassezia globosa on the occiput. We can get to the species level for Malassezia. We can also get to the species level for Candida. This was a study, an early study that we did, where we're now able also to obtain species level resolution of Candida. Important for the next part of the talk, we also with ITS1 sequencing looked at the different clades of Candida auris. So Candida auris clades 1, 2, 3, 4, and showed that there's enough diversity in the ITS1 region between the different clades of Candida auris that we can actually determine which clade of Candida auris we're obtaining. So I've kind of gone a little out of step, but I wanted to just start with the genomic sequencing and now take you into some of our studies that we've been looking at with Candida auris. As Judy was saying, that we're working on the human skin microbiome and emerging pathogens in Candida auris, thanks to conversations with my colleagues at CDC. They really brought me into thinking about Candida auris when it was originally being detected in the US. So this week, Candida auris has been in all the newspapers, but I'll still give a little bit of an introduction that clinical samples of Candida auris for bloodstream infections were identified 15 years ago. And at this point, starting about 5 years ago, we started seeing regional spread in Chicago and New York nursing homes. So for a long time, we've talked about antibiotic resistant bacteria, but in 2021, both the CDC and NIAID put azole-resistant Candida with drug-resistant Candida auris as an urgent threat. And that's because of the high levels of antimicrobial resistance, the difficulty in treating bloodstream infections. Patients remain colonized on their skin for months or years, and I'll show you that. And then patients shed Candida auris into the environment where it persists as viable. So I'm going to take you through a little bit of that, and then in our studies to look at Candida auris transmission. We developed a mouse model, in which here the staining is showing Candida auris on the surface of mouse skin after we've colonized mice. This is a mouse model where we purposely colonize the mice. We colonize the mice, and then a week later, we can find Candida auris inside the hair follicles of the mice. And so what we're seeing is sort of how Candida auris can become a permanent member of the skin microbial community. Just want to also highlight some really nice studies that were done on pig skin. And of course, this has now been updated with the most recent studies that launched the news reports, but really this concept that Candida auris is becoming endemic in several regions of the US. Okay, so skin biology. Your skin turns over just like your red cells. And we shed our entire outer layer of our skin every 2 to 4 weeks, right? So you've got basal cells and stem cells inside the skin, and you're turning over your skin. And that's probably a way to prevent yourself from developing any sort of malignancies because of the, UV radiation that you're experiencing. So we shed our skin, and that means that you're shedding 30 to 90 milligrams of skin flakes per hour. You're going to see those as the dust bunnies in your house. But along with that, really nice studies from Jessica Green and Brendan Bohannon lab showed that we also are shedding 1 million microbes per hour. And so in our houses, those become the dust bunnies, but in hospital and healthcare settings, those can become live organisms that are spreading, and shedding off your skin. So I'm going to take you now into our first study that we did in Chicago. And this slide is from the Illinois Department of Public Health, where at first they were doing surveillance. They identified 2 subjects who had Candida auris pretty early on. But really the change became when we started thinking about and realizing how much Candida auris can be a skin commensal and not just an infection. Look for that. Mihalis Leonakis, Diana Proctor, Rebecca Drummond, and I just wrote a review article about Candida and both commensalism and pathogenesis. It's going to come out next month. But really the model here is that when we are screening in these facilities, then we're identifying a lot more patients who are colonized with Candida auris, meaning that they're asymptomatic, but being colonized by Candida auris is a major risk factor for developing an infection. So the question was, why can't they get the Candida auris under control in this facility when they are starting now to do all this screening? And so in collaboration with colleagues at Rush University and the CDC, in this facility, in a study led by Diana Proctor, who's a brilliant NHGRI postdoctoral fellow and looking to complete her postdoc and go out onto the job market. So Diana worked on the study that she helped design, where the current recommendation is axilla groin sequence culturing for Candida auris, or surveillance for Candida auris. So as you've seen with our studies about healthy volunteers, we again did that, where we're looking at 10 different body sites, and Dr. Hayden is swabbing these 10 different body sites, and then in Dr. Hayden's lab, they go forth for culturing, which you'll see in a minute. And in our lab, they go forth for sequencing. And based on culturing, you're seeing the different positivity rates for the different body sites. And what we discovered through this deep analysis of 60 patients being studied on a monthly basis was that the axilla groin swabbing was identifying 68% of the positive patients. And as a result, what you can imagine that if you're only identifying 2/3 of your positive patients, then it's very hard to control the outbreak, because there's still 1/3 of your patients who are within the patient population who are continuing to spread. We found the NAIRS to be the most highly colonized site. And actually, so did the New York Department of Public Health. But there is still the question about whether that's clade specific. So I want to focus on the sequencing part of this study, and then to move into some of our other work with Candida auris. So really quickly, this is just ITS1 sequencing, where each of these bars is a different individual. There were 60 individuals, 3 time points. And the black line here is the amount of Candida auris that was detected. So sometimes you'll see other pinks, and those are other Candida. But we did ITS1 sequencing. We also did bacterial sequencing. And what this allowed Diana to do was, on a monthly basis, to think about what are the communities of Candida. And so based on the initial data, you can see that we could take every person and say that they had one of three types of fungal community. There's this cluster 1, which are people who have a fungal community dominated by Malassezia globosa, Malassezia restricta, what we would consider commensal Malassezia. Then there's a cluster of people whose body sites are dominated by Malassezia sleuthiae, a cluster of body sites dominated by diverse Candida, and a cluster of body sites that are dominated by Candida auris. And if you, Diana built the model based on month 1 and month 2 to say within that body site, if you have a Malassezia diverse community on the body site, there is an 80% chance that when you come back and sample that same site the next month, it will be Malassezia diverse. There is only a 2% chance that a body site transitions from Malassezia diverse to Candida auris. And so these models were built on time point 1, month 1 to month 2, and then tested to see if they replicated, and they did between time point 2 and time point 3. So based on that, Donna's showing that if you have a diverse Malassezia community, then likely you'll stay Malassezia diverse. But if you move from Malassezia diverse to Candida diverse, then there's a 38% chance that you remain Candida diverse, and a 38% chance that you would then move to being Candida auris dominated. And once you become Candida auris dominated, then it's also quite likely that you would remain Candida auris dominated. So that's sort of the version of that story, and that's published. So I want to just move on to talk about what we're doing now with these samples, which is that we talked about amplicon sequencing, and now we've moved on with this data set to look at shotgun metagenomics of these same samples and also the cultured isolates. Okay, I need to take a little detour and just talk about a breakthrough in the lab. And I hope I'll still have time to come back to Candida auris. I have not been going as quickly as I was during my practice talks. Okay, so I'll kind of move quickly then. When we use shotgun metagenomic sequencing, the method is that you remove the human reads, you take the non-human reads, and you align them to reference genomes, bacteria, fungi, viruses. And the problem with this is that the dirty little secret that people don't often talk about is that if you look at all our body sites, almost half of the reads are unclassified when we just use like a Kraken database. And that's because of many things, including that we don't have cultured isolates for many of the commensal organisms. So in a method developed by a wonderful, amazing, talented grad student who's now gone back to medical school, Sarah Saheb Kashaf, in collaboration with Alex Almeida and Rob Finn, we developed a method for metagenome assembled genomes, or MAGs, where you basically are going to create these genomes de novo from the shotgun reads, where it's kind of like as if you are so you take the sequencing reads, you assemble them as much as you can into contigs, and then you bin them based on characteristics of the genome, for example, tetranucleotide frequency, or also their relative abundance in the different body sites. So if contigs are from the same genome, they should be always at the same relative abundance, which might be quite high when the body site is the forehead and quite low when the body site is the antecubital. With this analysis, Sarah identified a lot of novel MAGs shown here in green, as well as MAGs that match to known isolates. And we tested this with isolates that were cultured, but SARS breakthrough here was not only in finding the bacteria, which had been done in the gut samples, but also to identify the fungi. And so cultured isolates are in red and mags are in blue. And of course, SARA recovered again mags that matched Malassezia restricta, Malassezia globosa, but also discovered 3 novel Malassezia. And really nicely, one of the novel species that we identified was then published as Malassezia aurunlochii. And that's just great because the mag matched the cultured isolate genomes like 99% identity, which gave us a lot more credibility that we could identify Malassezia this way. And I just wanted to draw your attention that in the supplemental materials, we do show how we QC these mags. We're basically here, I'm taking the consensus mag and I'm rerunning it where you can see individual samples and you can see that we have even coverage. And the bottom right would be an example where the coverage is not even, and we actually think that's a chimeric mag that's being generated. So there's a lot of QC that still goes into these samples. You can see that this Malassezia auris is found in more of the oily sites. So that's been, so. Okay, so the skin microbial genome collection, bacteria, fungi, viruses, how do we use that? Now we're using that to study these samples where we had the Candida auris and we had the skin shotgun metagenomics. And now we're going to look for, are we seeing transmission within the host or transmission between people? And we're going to use skin shotgun metagenomics and isolate sequencing. Okay. So I showed you this before, but I just want to remind you that this is our study. And now we're going to focus on individuals who are colonized with Candida auris. So we do shotgun metagenomic sequencing from the anus, fingertips, inguinal crease, which is the inguinal crease, well, it's shown up there, the nares, the toe web. And we're doing shotgun metagenomic sequencing and isolate sequencing as well. And these are the original culture plates. So this part I'm not going to go into. This is just sort of how we can find the different genomes and assemble these mags from this body site. But I wanted to focus today on the isolates that we were culturing from the Candida auris plates. So again, here you've got the isolates. And so like here you would have individual number 14, and we'd have an isolate from the perianal region from subject 14, month 1, month 2. You'd also have isolates from the fingertips at month 1, month 2. Sometimes if we're missing the inguinal crease, that means maybe that site just wasn't positive. The nares, we'd also have time point 1 and 2. And so we're using these isolates now to say, how similar are the Candida auris isolates between the different body sites of an individual and between individuals who share a room or are in the same facility. And so the analysis of the Candida auris genomes, we're taking the genomes and we're aligning them against a reference genome that we created from this outbreak in Chicago. And we also created a nanopore reference genome, so a closed finished genome, chromosomal level from the initial isolate identified in Chicago. We use an integrated pipeline of free-base BCF tools and GATK. And I'm going to punt that and say that if you want to know more about that, you should watch Christina Cuomo's Mycotalk. And she also has a paper in press that's really very specific to Candida auris. But what we're looking for are SNPs so that we can say, you know, are these two isolates more closely related to each other? Okay, so a lot of data here, but I'll walk you through it. And this is the final part of my talk. So here, this tree is every isolate from every subject in this facility that's I shouldn't say every, the vast majority of the isolates from the subjects who are positive in this facility. And we have the isolates color coded so that if it's from the same subject, they're going to have the same color. And we also integrated into this study published data from other studies in the Chicago area. So again, you'll see that those isolates cluster together. And here's examples of, in our study, subject 53, all of their isolates cluster together, 46. And that would mean that this person has isolates, has SNPs that are specific to them, that you can see that they are, all clustering together. That might suggest that you would be able, so the thing is, you want to have enough diversity that you can maybe track to see diversification within an individual, but what about within a facility? So we looked at this quantitatively and looked at how many SNPs do we see within the same subject versus comparing different subjects. And you can see that when it's the same subject, there's this hump over here where there's very few SNPs between different body sites. But there's also the second hump here, where between different body sites of the same individual, there can be as many as 20 SNPs, which is the same as what you're seeing from different subjects. How would this be? You could imagine that someone could be colonized in their nares with one strain, but they're walking around and the isolate that you're picking up from their toe web or their fingertips is representing what's in the facility rather than what's on them. And that's in fact what we're seeing here, where you're seeing the isolates from the nares of this subject are mapping here in the tree, whereas the isolates from the toe web are mapping to a different site. Sometimes the inguinal crease isolate is mapping here, but on next month it's mapping here. So there can be quite a lot of variation across space and time. And that's going to make it very difficult for us to use isolate sequencing to identify transmission networks within facilities. But we do look at it in the context of, can we explain this, that the person, in this case, lives in room 1 of the facility. And what you're seeing here is that the subjects who live together in room 2 also have isolates that map together. But you can see that the subjects who live in room 1 will sometimes have multiple different clusters, suggesting that even within a room, there are multiple different types of isolates of Candida auris that are in that room. So in conclusion for my talk, I just really wanted to go through some different flavors of how we are characterizing microbial communities with isolates, with shotgun metagenomics, with amplicon sequencing, and then to finish by saying, really, where is the future of the lab and for us? And I would say that a lot of this is that genomics derives hypotheses, but that then will utilize isolates cultured from samples to go forth and test those samples, to test those studies, to test those hypotheses. And I know others in the community are doing similar types of experiments, like with Candida auris. One of the hypotheses that you saw with the model was that there is a Malassezia diverse community is less likely to be invaded by Candida auris. So are we seeing ways in which Malassezia can help to provide colonization resistance and inhibit Candida auris from coming in? So there, what we've been doing is really just storing a lot of isolates from subjects and trying to use isolates that are all from the same individual to test hypotheses generated with genomics. In conclusion, I'll just like to really acknowledge all the amazing people in the lab. In particular, I focused here on the work of Diana Proctor and Sara Saheb-Kashef, but the work on the Malassezia and the Candida is now being spearheaded by Jonathan Nicholas, and now I'm sorry to Jonathan. I realize I must be having an old slide because I didn't have him included in here. Apologies. I'll update my slides. And the fabulous collaborators that we have here at the NIH, Mihalis Leonakis and Yasmin Belkade, and at the CDC, Anna Litvintseva, as well as my colleagues at Rush, Mary Hayden, Mike Lynn, and Mike Shoney. And with that, I'll finish off and turn it back over to the meeting organizers.
Great. Thanks, Julie. Wonderful talk. So our pleasure to introduce our next speaker, Dr. John Perfect. Dr. Perfect is the James B. Duke Distinguished Professor of Medicine and Chief of the Infectious Disease Division at Duke, as well as Professor in Molecular Genetics and Microbiology. Dr. Perfect did his medical training at the University of Toledo, followed by an internship at Riverside Methodist Hospital in Columbus, and then a residency at Michigan, and then a fellowship in infectious disease at Duke. And there at Duke, he's launched a program focused on Cryptococcus neoformans, and has used this as a model organism to study cryptococcal virulence factors and genetics using fungal transformation strategies, but is also to try to identify novel targets for novel antimycotics and also immunotherapeutic strategies. And has recently been awarded an NIH-sponsored mycology unit to support multiple investigator studies in Cryptococcus. So Dr. Perfect's going to talk about carbohydrates in the cell wall, I believe. So looking forward to talk.
Great. Thank you, Jay, and thank you, Julie, for inviting me to talk on this this subject. I want to talk on the sugar-coated killer yeast, which is actually Cryptococcus, which I've studied for about 45 years. And what I'm going to try to do today is talk about three areas. Number one, just the epidemiology on it. Number two, some of the basic science around it. And three is looking at the issues around guidelines. And so I'm going to go pretty fast to stay on time, but bear with me as we go through these slides. So again, it's my pleasure to do this. This is the beast. This is the organism that we deal with, the encapsulated yeast. I'm going to spend a few minutes on a cohort that we actually did at Duke University, where we retrospectively looked back at what we did with Cryptococcus. We are a pretty high incident area, and we did this a few years ago, and it may need a little updating, but in some ways it probably is what we deal with today. And I want to emphasize on this thing, a little thing is there are three groups that we put this in: HIV positive, transplant recipients, and non-AIDS, non-transplant. And the important point to this particular slide is how fast we can make the diagnosis of crypto from the time of symptoms. And you can see for HIV positive patients and transplant recipients, which are all known to be a risk factor for Cryptococcus, it's still, about 3 weeks before we get it. But look at the HIV negative and non-transplant patients. It takes an average of almost 50 days. So this is an important issue for us as clinicians, how fast we can make this diagnosis in patients that start to have symptoms. The second thing I noticed a little bit over time was our attributable mortality in the HIV positive patients, which you see here, versus the transplant patients versus the non-AIDS, non-transplant patients, very similar mortality rates in the HIV and transplant patients. But in the non-AIDS, non-transplant patients, it's much higher. And that's not surprising, I think, in some ways, because it takes longer to make the diagnosis. And the IRIS, or the immune reconstitution inflammatory syndrome, is in each one of these major groups of patients that have cryptococcal disease. I think when I looked at it over this time, the influence of initial therapy on outcome for Cryptococcus, I'd like to emphasize 2 points. One is that flucytosine, a drug that we use with amphotericin B, was associated with a lower overall mortality. So I think that combine in using flucytosine is really important to the outcome of these patients. And unfortunately, most of the world does not have access to flucytosine, which is unfortunate. The second thing is receiving a non-recommended 2012 IDSA guideline regimen had a higher relative amount of persistence of disease and rate of attributable mortality. So I think the guidelines that we have out there right now are helpful to the clinicians. You can't always follow them, but you should start with them as your baseline. And I think the outcomes would be better. I want to put an emphasis up here that what Joe Jarvis did with cryptococcal meningitis. And you think HIV is under control, et cetera. Well, we do have pretty good drugs, and the amount of cryptococcal disease was pretty high in 2022 and 23. But actually, the HIV patients, despite ART therapy continuing to increase, has leveled off. And so they can still do quite a lot of studies with very large numbers of patients, even today in the era of really good ART therapy with the incidence of disease. So unfortunately, we've made some impact, but there's still quite a bit of disease. And for us in the United States, we have small molecules and stuff like that that we use today that have increased the incidence of Cryptococcus. And in our own hospital today, we probably see not quite as much as in the AIDS epidemic when it was at its highest, but still very frequent, 1 to 2 cases of cryptococcal meningitis a month. I wanted to emphasize here the outcome of these things, the average cost to cryptococcal meningitis when we looked at this. And again, it's added cost to a hospitalization is over $20,000. But look what happens over the 5 years. You eventually get up to almost $100,000 increased cost for patients that have cryptococcal disease compared to groups that have similar types of underlying diseases. And again, upwards of 50 to 60 days of hospitalization. So the impact of cryptococcus over a 5-year period is still substantial, both in money and in time and effort. Now I'm going to switch gears a little bit. I'm going to talk about the organism, a little basic science. We're going down into the beast. It was actually for us, H99 was born on February 14th, 1978 at 11 p.m. at night. This is an organism I worked with for over 40 years. And you say, well, Dr. Perfect, I don't really believe that. You're just making a guess on that. And actually, no, this is my note from 1978, 11 p.m. at night. A gentleman with Hodgkin's disease had cryptococcal meningitis. His opening pressure was over 400, so he had an increased pressure. His glucose was low, so hypoglycemia, and he had some cells. And we started him on standard therapy back at that time with pretty high doses of flucytosine and amphotericin B at pretty low doses, and when we just increased it over time. What I like to say is he was treated for 6 weeks and he was cured of his infection. So again, H99, an organism that we've now used extensively in kind of the type culture for much of the studies. This is all where it started on that day, Valentine's Day, many, many years ago. My wife's still trying to figure out exactly what we were doing on Valentine's Day in 2/14/78, and I was in the hospital trying to take care of a patient and seeing H99 for the first time. I want to put an emphasis here that these organisms do change, they can evolve, and even H99, you can see that it has phenotypic variations, and this becomes important as we study this organism. We'll put up here the rabbit model. This is quantitative counts over on this side and days of infection. We intercisternally inoculate this organism and we use steroids to suppress them. And this is our H99, what we call stud, or the most aggressive, coming directly out of that patient in 1978. But this is the organism that stayed around and went through many, many labs throughout the country and around the world, and eventually came back to us with it actually mutated with a gene called LMP, low mating potential. And it turned out that this organism changed. And you can see here how it dramatically changed in the animal model to become a virulent. So again, these things and these organisms can change. I just want to put up there that change can be rapidly. We know about karyotypic instability, heteroresistance. I've had changes in copy numbers as we follow these infections over time. We now have over 2,000 cryptococcal genomes sequenced, so we can actually start to interrogate the evolution of these things as they change over time and from different patients, and both in the environment and not. I've used an animal model for many years, which we inoculate directly into the CSFs of rabbits, use steroids like patients. And you can see here that it acts, the control infection does not go away. Eventually, the animals would die for this infection. Fluconazole has a more fungistatic type of area, and then amphotericin B looks very, very good and very consistent. And I'll come back to that in a moment. So Cryptococcus is going global. Does it really tell us anything? And so we used a lot of gene expression, having the organism try to tell us what happens during infections. And in our early studies, we thought an organism like isocitrate lyase was highly upregulated in both animals and in humans and crypto. But when we knocked the gene out, it really had no impact on Cryptococcus, unlike with Candida. On the other hand, we saw high levels of expression of the trailose pathway, and it's been an area that we've been able to explore and understand its importance to the organism's ability to produce disease. So the power of these expression studies are actually enormous. It allows the organism, the yeast, to directly understand or help us understand what makes it tick, and potentially allows us to study undefined genes, so that we don't go on just what we can see and make hypotheses on. We can actually look at these things and say, this is what's happening in the patient. So life, like life, in the global arena, there are a lot of choices. Choose wisely. Hopefully you don't choose poorly. And we've tried this over time and have done both. This is where we do the CSFs of patients. It's directly taken off the cerebral spinal fluid. We don't put it on culture plates, so it goes directly in, and we isolate RNA directly from the organisms coming out of the cerebral spinal fluid, trying to understand what their transcriptome actually is. What are they thinking at the time of this infection? And we now have over 35 patients that we've actually done this on. So we have a pretty good idea of how the organism is thinking in vivo, not in a plate, not in all these other things, but actually in the host. So you can see up here the networks that we can actually describe. This is a network of looking at YPD grown cells versus coming directly out of the human CSF. This is a rabbit CSF versus human CSF. And this is the organism changing over time within the rabbit as we can follow it serially over time. So you can see there's some dynamic relationships that are going on. We can pick some of these things out, and we can find genes that, like this particular CMP gene, that was highly expressed. Okay, well, what's it doing? It must be important. We knocked the gene out, and you can see both in mice and in rabbits that it is important to its ability to produce disease in both the lung. And here, actually, this was males, and these are females. For everybody that does NIH grants and stuff like that, this is for you that we actually. There was not a sex difference between the two. And you can see in the CSF, it was important once you got rid of this CMP gene. Again, we don't know exactly what this gene is and stuff like that, but we now have the idea to explore it in relationship to other genes and other pathways. Same thing when we do sequencing. We've now sequenced over 2,000 to 3,000 strains. We can start to look at strains and their sequencing, do GWAS type of analysis. And in this particular one, we actually took a group of organisms that actually had certain sequence differences in the 4102 gene, which we don't know exactly what it does, but there was a group of them that were associated with high burden of organisms in the CSF of patients. And then we took the ones that were more wild type. And you can see here in the rabbit model, we kind of copied this type of setting, that actually the ones that were higher concentrations in the humans were actually had more mortality in the mice situation. We also saw in the rabbit situation a gene that was associated with seemed like a very aggressive disease. If we knocked it out, we could see that it had less activity than the wild type strain, and even over time, you could see that when the mutant was less virulent or less aggressive than H99, our standard strain. So we can do all these things. We can pick these things out. We can take them back to the animal model and try to dissect what happens. It's a big world. There's a lot of sequencing goes on, et cetera, et cetera. But we've done situations where we looked at strains over time in patients that relapse. And so we can see the mutations occur or the changes occur in vivo. At this stage, we're looking at a lot of transposon type of activity that really moves around as these organisms stay in the host. As I mentioned, we got many genomes that we've sequenced, and we've looked at clinical isolates and looked at cell size and phenotype associated with mortality. And as I mentioned, we can now look at what the organism is trying to think in vivo, coming directly out of the patients by doing RNA-seq analysis. And now we have a series of pathways that seem to be important to this organism's ability to produce disease. We have a sequencing of this thing. So now we've done environmental and clinical isolates. We have them in all types of categories and put together in kind of a phylogenomic analysis. What I will tell you, and it's not shocking, but that in fact, the clinical isolates, when we put them in animals, we've done over a dozen of these patients coming, these isolates coming from the clinic, or not clinic, but coming from the environment. And then we take the clinical isolates that have been in patients. There's dramatic differences in survival of the mice when they're infected with one versus the other strain. The environmental strains are generally not as pathogenic as the clinical strains. This is not shocking. The tools and the selection of the yeast that cause disease in humans is well reflected in the ability of very controlled situations under mice infections. Now, I'm going to just make a couple points on 3 experiments in clinical practice of cryptococcosis that I think have some relevance. The first is we've been studying with Mycotalks neurophoresis. So we're actually dialyzing the subarachnoid space. And you can see here in a rabbit model, we put the catheters in here, here, and there is a filter here that can filter out organisms like Cryptococcus. This may be extraordinarily important in the early infections where you get blockage and increased intracranial pressure. And so we've been doing this over some time. And actually, the other thing we could do is actually put antifungal agents like amphotericin B in this whole cycle. We've done this over time, and actually we can do this. We can knock the burden of organisms down in this particular situation to almost 2 logs in 6 hours by dialyzing through the subarachnoid space. Is this going to be important to reduce the amount of organisms and debris and stuff like that from patients that get increased intracranial pressure and stuff? I don't know. That particular system is by Mycotalks, and it actually is trying to get approval for subarachnoid hemorrhages and is close to actually doing that as we talk now. Hopefully, if they get that, then I can see a scenario where patients with increased intracranial pressure, a lot of organisms sitting in the cerebral spinal fluid, blocking the arachnoid villi. We could actually dialyze those cells off with this kind of system. Surely in the rabbit, we can do it. Now, the next thing was a student, Blake Sells came. He was an undergraduate student. He was reading about a bunch of stuff, and he says, "Oh, Dr. Perfect, ketogenic diet. I wonder if that can really help your antifungals and stuff like that." And I says, "Well, Blake, your thought process is kind of nice, and maybe we can get interaction here. And because you're so enthusiastic and stuff like that, I'll give you 40 mice to actually see if a ketogenic diet in combination with fluconazole can lower the tissue burden in mice." And the answer is he was absolutely right. This is untreated on standard diet. This is on a ketosis diet. And then treated here for fluconazole could knock the organisms down a little bit. But oh my gosh, on a ketotic state, you can see the dramatic 3-4 log difference with fluconazole, suggesting that this was very dramatic in knocking the organisms down in the brain. And actually, this also occurred similarly in the lung. So something was happening under the ketotic state that allowed fluconazole to become a fungicidal agent. And pretty dramatically so. We've repeated this multiple times. So we tried to figure out why in the world that would happen. And so actually it turns out fluconazole concentrations are a little bit higher in the ketotic animals. And you can see here that the area under the curve is a little bit higher. There's no question about that. And so the tissue levels may be a little bit higher for fluconazole, but they could never be in the kind of situation that would have that kind of dramatic in vivo effect. So there had to be something else. We then went through and looked at things like the small chain fatty acids abundance in brain tissue. And it turns out under the ketonic diet and stuff like that with fluconazole compared to without fluconazole, the regular diet, there were more of these small chain fatty acids in the tissue. And that became important because we looked at butyric and hydroxybutyric acid. And it turns out that actually butyrate actually has an MIC to fluconazole of 2.75 and not as high for hydroxybutyric acid. But again, some activity. So all of a sudden, some of these things related to ketosis may actually help and synergize with fluconazole. And that's actually true. In vitro wise, fluconazole and sodium butyrate together are synergistic. This is an FICI that's way under 1, and a synergy between butyric acid and fluconazole that might be showing the potency that we actually see in these animals. So we are in the process of continuing to dissect that. The final thing is with GT1 inhibitors, new kind of class, inositol acylase inhibitors that clearly look at GPI anchored cell wall biosynthesis. And one of the drugs in clinical studies is APX001, which is phosphomanogiopex. And its MIC is okay, but they had a series of other congeners. And in there is a drug called, or a compound called 2039. You can see the MIC are dramatically lower at 10 to 100 fold. And this is the organism before it's the strain, not strain, but the compound and kind of pre-drug that is broken down to 2039. It had tremendous kill and sterilization of the tissue. Okay, well, that's fine. Does that make in vivo type of activity? And you can see here that actually this drug, 2039, this is a compound that keeps it so it isn't metabolized as rapidly in animals, but dramatically different than our fluconazole and amphotericin B, significant amounts of reduction of organisms in this model in mice, colony-forming units in both the lung and the brain. And this is in the CSF in rabbits and stuff like that that we actually do. And I'd like to emphasize this dosing that we actually have here. This particular compound by far is the most potent compound I've ever studied in any of the compounds we've ever had. Much more effective than amphotericin B or fluconazole. And it actually has a dose relationship. So we know where we need to hit to get unbelievable amounts of killing. And in fact, in our animal model, in our rabbit model, which is this quantitative units in the CSF, we can actually get sterilization of tissue in both the brain and in the CSF in less than 2 weeks of therapy. I have this thing in the back of my mind that someday a drug like this, you could give oral drug and treat patients for 2 weeks instead of the 6 weeks or 4 months or 6 months or a year that we actually do today. Incredible potency of this drug. Okay, I'm going to finish with the last 5 minutes or so, going to the third part of this thing, which is my exposure to leading the guidelines. I had the opportunity to lead the IDSA guidelines in 2010, and recently we finally finished the European guidelines in 2023. They will eventually come out. What I know about guidelines is they are a lot of work. This particular situation, because of COVID and everything else, has been 4 years in the making, but we're really close and should be staying in the next month or two. Without substantial infrastructure, final product could take a long time. And guidelines are static and clinical practice is not, it's dynamic. But there's a balance is needed between data and opinions, but we need to put them out there because even though no patient I know of is really, well, very few patients have ever read the guidelines. It's good for everybody to be on the same page. And I think that these guidelines that will come out shortly will get everybody on the same page, both on a local, regional, national, and international perspective. Just look whenever you see these guidelines, some of the things may be in the supplemental area because the guidelines took about 115 pages to actually get down, and you can't do that in the primary guidelines. So you'll see a lot in the back. The clinical practice guidelines for the management of cryptococcal meningitis. Cryptococcal disease was 2010. This was the IDSA ones. I learned a lot from this, I think, and I think they're still not bad, even though they're almost 13 years old. But I would want to emphasize one thing, and the abstract in that particular paper is still as good today as it was back then. If the diagnosis is made early, if clinicians adhere to the basic principles of these guidelines, and if the underlying disease is controlled, then cryptococcosis can be managed successfully in the vast majority of patients. I still believe that. And let me show you a little bit of data on that. Now, the new guidelines, the European Committee on Medical Mycology, led by Oliver Cornley, but we've had upwards of 40 patients participating, and I would like to do a specific shout out to Christina Chang, because she's just been outstanding in trying to put all this thing together. She's come back and forth to me. We've been doing this kind of, the two of us, for the last year or so, and I think it's coming to a really good place. What I want to emphasize a little bit about this is the cryptococcal meningitis treatment. One of the things that's very important is induction therapy. I'm putting on my clinical hat right now, and the guidelines suggest one of two ways to go. Liposomal amphotericin B, as you can see on the dose, and flucytosine. That's done for 2 weeks. Or a single dose of lipid amphotericin B, or liposome, or ambisone, and then 14 days of fluconazole and flucytosine. This is the most recent study, the Ambition study that was published last year. And this has been a tough problem because, again, the issues around what is best for resource limited areas versus resource available areas is not clear. One of the sad parts that I see is all this was done in Africa, or in low resource areas, which is absolutely fine. That's where most of the crypto is at. But does that give the same impression and the same success rates in resource available areas? And that answer we just don't know. So these are the recommendations are one of two. I want to put up there that if you look at that and the effect, the short Pauling induction therapy in that particular study last year in the New England Journal, the 10-week mortality rate was 25%. If we look at a standard Pauling induction regimen in the United States of the 2 weeks of the ambisone and flucytosine, mortality is variable, but up to 1 year, we have two studies. We did one and somebody else did one, looking retrospectively back at big databases, that between 6 and 12% is the most recent mortality rate in looking back in this kind of 2-week regimen with Pauling. And in fact, a group out of Japan just recently published, who again with this standard 2-week regimen, a mortality rate of 6%. I don't know. Is this different? Are these two different? We don't know, because we have not done this kind of regimen in resource available areas. We simply have to figure out and do the studies. I want to emphasize again the screening and primary prophylaxis for preemptive therapies. Things we did in the guidelines and stuff like that. You can see we're now screening patients that have low CD4 counts with CRAG. Positive ones need a complete screen. Those that are negative are put on fluconazole at 800 to 1200 milligrams. There's been several breakthroughs of this type of situation, and therefore azole compound has become less and less exciting to be used by itself. However, what are you going to use? And so there are now 2 studies that are trying to optimize empiric therapy, and they're in progress. One with fluconazole and flucytosine in combination, and the other with intermittent dosing of ambisone. So we'll see what happens over time to get to these patients very early before they actually develop severe cryptococcal disease. And then finally, some places have tried primary prophylaxis, but I think with the antigen testing system today, there are a few places that need to be doing prophylaxis, primary prophylaxis, that most of them can do some type of screening type device that we talked about. Okay, so I'm going to leave, and I think I got a couple minutes left. I haven't seen that 5-minute thing come up, but I want to put up a couple question issues that we've had to deal with now over time, and they're in the new guidelines. Should we elevate lipid formulations of amphotericin B over amphotericin B in resource available environments? The answer is yes, and I think it's just standard of care. In non-HIV and non-transplant patients, should we encourage prolonged induction time? This is our group that has the highest mortality. In the past, a lot of people have used 4-week type therapy, but we've used 2-week type induction therapy in transplants and AIDS patients fine. And in fact, in the guideline, except maybe for Canada, Cryptococcus gattii infections, 2 weeks will be standard for all patient groups. With lateral flow assays, should we aggressively rewrite an integrated strategy for antigen detection and treatment in resource limited environments? The answer is yes, but it's ongoing as we talk and studies are ongoing. Some people do this in high incidence areas, and what the best regimen is not absolutely certain yet. Are the broad recommendations of 2 to 10 weeks for heart after beginning cryptococcal induction in need of revision? That's a really interesting question. We've gone on the 4 to 6 weeks like the COPE study suggested, and we did not do the early ART therapy and early administration during the treatment of cryptococcal meningitis, but delayed it. There are a recent study, a recent paper in the US and resource available areas that suggested you might be able to start the ART therapy a little bit earlier when you're starting therapy for cryptococcal meningitis. But if you do, you're going to have to be very careful watching these patients. Should corticosteroid therapy in cryptococcal meningitis be readdressed? Well, I think it's been addressed multiple times, and I think for iris, it's still part of the thing, but they have actually done this thing with the DEX study that showed that in standard, just using steroids with cryptococcal meningitis right up front is probably not a good thing. It's done in the patients that have iris and inflammation, and there again, it's tricky, but it should be instituted. Should in vitro testing be encouraged in all isolates? I actually think it should. I think we should know, although there really is very little drug resistance a priori today. It's when you get the relapses, are they resistant or not? Should we consider quantitative yeast cultures? These are EFA type of measurements, which are done in many of the studies today. Your labs are not going to do these things, no matter how much you do. So I think it's great for clinical studies. It's probably not going to be done on Main Street. Have we done enough with increased intracranial pressure recommendations? We tried very hard in these new recommendations to give you all kinds of ways to do this. But again, good studies to really show you exactly how to do it will not be clear, and they have to be done at the bedside, but you need to pay attention to increased intracranial pressure. Are C. neoformans and gattii treated the same? Actually, right now, it's very hard to separate the two out, and I think for practical purposes, they're treated the same. In resource-limited settings without flucytosine, should the combo of amphotericin and fluconazole be elevated to primary therapy? Tricky issue, but I think probably it should. Should we recommend timing of follow-up LPs? Those are tricky issues, but one should actually get a follow-up LP probably at least at 2 weeks.
John, if I could just give you a notification, you have 1 minute remaining.
Oh, okay. I never saw it. I'm sorry. Thank you very much. I'll finish up. Should we integrate with WHO guidelines and up-to-date? We are trying. I write the up-to-date and the CDC guidelines, and we're trying to integrate with the new guidelines that will come out the next couple months. And finally, Cryptococcus, a sugar-coated killer. It is important, it's deadly, and its mysteries are being unwrapped. In treatment, the goal was efficiently killing the sugar-coated yeast. Fungicidal targets, fungicidal regimens, we got to do that. I think the pathogenic principles are set. Weak spots are many, but the relationships are complex and dynamic. Thus, keep close to the host. Control for change, analyze and choose wisely from the massive bioinformatic data that's now being generated today. We need to realize it's the host, it's the yeast, it's a two-way street. But today, more than ever, we can start to understand what makes this organism tick. And there's a worry, there's a goal that in 2030, it will be eliminated as a significant disease. I don't know whether we'll do that. That's what's been suggested, but we will know a lot more about this organism and the ability to control it. So this is my group. This is the group that does all the work, and I put them up there as so important to the ability of understanding this encapsulated yeast. So first of all, I want to again thank everybody that was on this thing. I thank people for the invitation. I know I went fast, but it was the ability to give some enthusiasm about this sugar-coated killer. I think we are getting better and better at managing it, and it's not going to go away. So thank you.
Thank you, John. Great talk.
Thank you
So
very much.
Yeah, so I'd like to turn it over to Dr. Berman, and we'll start the Q&A session here.
Yeah, so we'll start with some questions for Julie. First of all, Julie, I have a question from Ted White, which I'm going to then combine with one other one. Fungal cell wall can be hard to disrupt compared to bacteria. So he wants to know what lysis buffer you use and whether you're concerned that the amount of fungal DNA in the sample is proportionately less than the bacterial DNA. Are the fungi being underestimated compared to the bacteria in the samples? And have you done any reconstructions to test that? And then along with that, what's the prevalence of filamentous mold on skin when using either ITS1 or that's probably the way you would do it? Yeah.
Yeah, thanks. So I was very fortunate that Keisha Finley joined the group from Joe Heitman's lab in 2012. And together with the biologist in the lab, Clay Deming, we did a lot of those controls where we created mock communities. And I should have highlighted this more. We did that, and I showed them on the slide that we did them as DNA samples to realize what kind of if there were any discrepancies in the ITS1 sequencing. But as you rightfully say, we actually started with doing those with live organisms and had Malassezia and some Candida and Saccharomyces cerevisiae. And we take our skin swabs in with a foam swab that's been moistened in the yeast lysis buffer. And so we then did, from organisms, we did our standard DNA prep. And I think the important thing there is that we do a high temperature bead beating step. And we also, and that's not a standard part for the people doing microbiome studies that are studying bacteria in the yeast. But it definitely, for most of these studies, you're going to do very short sequencing, a couple hundred base pairs. So honestly, why not just beat the crap out of your cells? Because you're not really trying to maintain large fragments. Yeah, we did a lot of those kinds of studies, and I think we did try to bury at least some of that as our mock communities in the supplemental materials to Keisha's original paper. And I would highly recommend that other people do those things as their positive controls as well. We also added into those kinds of mock communities, we added in the most common bacteria to make sure that when we did this high temperature bead beating, we weren't changing the bacterial communities, and we weren't. But I agree with well, of course I agree with Ted, that it does make me a little worried that for a number of the studies, they haven't released the fungal DNA. And of course, if you don't lyse the cells, then you're completely blind to something that you're missing, which is the fungi in the community. So all of our studies are done, even if we're doing something where our goal is to look at a bacterial community, we're always doing it with the same DNA prep with the yeast lysis buffer, thinking that those are the harder cells to break open. And again, it just comes back to really having to think deeply about what might be the ways in which you're not seeing things when you're doing these genomic studies, and as best as we can, with great advice from mycology colleagues, to try to set up mock communities and do those kinds of analyses so that you could have more certainty about what you're not seeing if you aren't looking for it.
So to follow up on that, do you see filamentous molds on the skin when you're doing the ITS1 analysis, or is the ITS1 not designed to see them?
So I would say, then I'm going to defer to my fungal colleagues. I mean, I I think in this case, I wouldn't, I mean, I wouldn't expect, like, if you're thinking about, I guess I don't know that someone has looked at that in the skin. In the gut, like if you're looking at Candida albicans and you're looking at the hyphae form versus the yeast form, I wouldn't expect that you wouldn't be able to lyse it and get an ITS1 sequence. But the filamentous, but you've now exceeded, like, I don't know an example of something, and probably you all do, an example of something that I should be looking for, and have I ever seen it? I just don't know the answer to that. Maybe someone can tell me like an example of what sort of filamentous mold you would be looking for.
Okay, we'll come back to that, and I'll let Jay ask a question of John in the meantime.
Great, thanks. Yes, we have a couple questions around the ketogenic data, which I thought was also very intriguing. So, John, the questions circulate around the clinical translation. So how long does the ketogenic diet have to be placed, and then does the ketosis have to be maintained during the treatment protocol? Like, so, is this something that we could even think about transferring clinically?
Yeah, really good question, Jay. Boy, you got me scared how long to do it. I'm not quite ready prime time to put it in a patient, but it has been very consistent in what you saw up there in multiple experiments. And actually, we started them a month ahead of time for the mice and put them on a ketogenic diet, and they were definitely in ketosis and stuff like that when we did it. It actually probably doesn't take that long, and of course humans may be a little bit different than mice and stuff like that. So we started a little bit ahead of time, and we did maintain it over the entire time because I didn't know how it would play. We didn't stop it in the middle of the treatments and stuff like that.
And have you had the opportunity to see if have you had opportunity to work it? Would it work, do you think, in a T-cell deficient animal like a rag mouse?
Good question. I don't know the answer to that, but my gut feeling is that it will.
Okay, that's exciting.
I don't think we're in the process actually doing a series of immune studies, Jay, right up your alley and stuff like that, to see if there's any impact on immunity on these things. I don't have those results back yet. I was so much focused on making sure that what we saw was real, reproducible time and time again. And
yeah,
it
exactly.
is. And so if some of your experiment thinking about rag mice or cytokine wise and all that we do on these things is actually in the hopper right now. The experiment's ongoing, and I don't know the answer to that.
Right. And then one other quick question from Ariel Butts is about the comment, or basically what's the impact of the intracellular macrophage crypto on treatment length and success? Is that an important stage of infection where the fungus can essentially escape antimycotics and then potentially either result in persistence or recrudescence of infection, I guess.
Yeah, I think it's an important issue. I've always looked on Cryptococcus as both intracellular and extracellular. It's both. And I think you do have to pay attention to sometimes to the intracellular compartment, because that may take much longer to get rid of than the extracellular. So when I say there and I'm doing CSF and pulling them off and doing quantitative counts and saying, oh, they're all gone. Well, that's not inside the tissue and stuff like that where the organ is at. But I would say that drug, the drug 2039, I don't know, maybe, but it was not just the CSFs coming out of the rabbits that were sterile. We sacrificed the animals at the end and chopped up the whole brain and went through, and there was nothing growing there. So those were being killed probably also intracellularly. But it's a good question. I think it's a combination, and you got to be careful sometimes that what looks on the surface killing in fluids and stuff like that is not what's being killed in the macrophages.
Great. Thank you. I'll turn it back over to Dr. Brimman.
Yeah, so Laura Roche brought up a question about the isolates that you described from the Chicago outbreak that have different SNPs in them. And the question is whether there's evidence that some of these isolates are not related to the outbreak, but colonized people separately, or did the organisms evolve after colonization?
Great, thanks. So backtracking, because remember I told you I knew a lot about genomics and about skin biology and less about microbiology. But so I just looked it up for filamentous molds. And yes, we find Aspergillus versicolor and Tinea versicolor on the skin. So Aspergillus versicolor, we've cultured and sequenced from subjects. So I would say, yes, I wouldn't see any reason that wouldn't be applicable. Okay. Now, the Candida outbreak in Chicago, we have the original isolate from 2016 and are still in touch with Illinois Department of Public Health, Chicago Department of Public Health. We believe it's a regional outbreak. And we basically can still link back the isolates that we're culturing and sequencing back to the original strain that came to Chicago in 2016. When we sequence the isolates from Chicago, we can also see other isolates that have been sequenced and deposited. And what you see there is that there will be case reports of the same clade that we find in Chicago being identified in other states. So there's a published report of a patient in Massachusetts with a published sequenced isolate that very closely matches what we're seeing in Chicago. And they also report that this is someone who is from Chicago who went to Massachusetts for their treatment. There are 2 isolates in Florida, and those are hundreds of SNPs different from the Chicago isolate. And there is no epidemiologic link to Chicago. We really can't explain it because there are some states where you see multiple clades of Candida auris coming in, and there could be multiple introductions that, Laura's asking an astute question that one of the reasons for this is asking about, are we seeing new introduction into the region from other states or from other regions? And so far, we just haven't seen examples of that. We still see that we are having a regional outbreak in the Chicagoland area. And as we saw during COVID, a lot of these patients are living in nursing homes, but they're entering the healthcare ecosystems and entering, hospitals and moving sometimes even back into back into other healthcare facilities. But at this point, I only have evidence for regional spread within the Chicago area. But there are, of course, New York, New Jersey is also having a similar regional spread. But states like California would have multiple clades coming in simultaneously. Really there, the important part is to detect Candida auris early and try to get it out of your healthcare system before it becomes endemic.
Okay, thank you. Jay?
Yeah, so John, we have a question for Joanna Canario about, so you showed data with the GWT1 targeted molecule, but what do you think the most promising drug targets are for crypto, and what does your crystal ball say in terms of potential upcoming clinical trials with some of these agents?
Yeah, Jay, so really a great question. The crystal ball is always fuzzy on these things, but I gave you the single best drug possible for Cryptococcus in 50 years of studying it. That drug 2039 in that class, better than the phosphomanogiopex or the manogiopex that's actually being studied, is as good as it gets. And I think it's just a matter of getting it in the right place. It's owned by Pfizer. This is not going to make Avogadro amounts of money, but it could be studied very quickly, particularly in crypto where you got Africa and places like that you can do patients, 200-300 patient study in a year and a half. This is not something like the other fungi you have to deal with and you struggle for years to get enough patients. So it's a matter of the company wanting to do this or selling this or getting somebody to do it. I don't need to search for other drugs and stuff like that when I have one that is actually probably pretty safe in a good class and we know where it's at. And I would say the rabbit model has pretty much mimicked what has happened in the humans. I would be shocked that it will not be the same way. And what do you want? You want a drug that maybe can cure a patient in 2 weeks and it's oral? I don't know. It's as good as it gets. So I'm sitting on something I'm kind of frustrated with a little bit because I think it's the best that I've ever seen. And right now, no one wants to deal with it, particularly Pfizer.
Very interesting. And in the pheresis model, do you also see a drop in the intracranial pressure in the rabbit model along with the CFU?
Well, you should. It was so hard to set that up. It's so ticklish and stuff like that with surgeons and everything else that it seems to drop. It does seem to drop. But I'm not going to tell you exactly how much it drops. But the thing about it is we can control that. You can control that. Actually, once you got that circulation going, stuff like that, you can bleed stuff off. And that's an important issue because as these people are asking these questions, this thing might be very attractive in those early stages with high burden of organisms. And remember, it's not just killing the organism, it's trying to get it out of there so it's not blocking up the arachnoid villi and stuff like that. And this is better flow. And so I actually think someday, if they get it in for subarachnoid hemorrhages, that eventually this thing could be used in patients like cryptococcal meningitis, where surely they have outflow obstructions from all the yeast plugging up in the arachnoid villi and stuff like that. So I think it still has very good potential. It's just got to make that next step and actually get approved for a more common disease.
Great, thanks.
All right, so Julie, there's 2 questions left with sort of more molecular ideas. One of them is whether there are factors, whether you know of the factors for malassezia that may be inhibiting Candida auris colonization. And the second is whether diabetes patients have more colonization, which is known for albicans, but what is known for auris about that?
Yeah. Okay. So the question about the malassezia and the Candida, that's sort of what I was trying to get at with my final slide of saying that genomics is generating these hypotheses, but that we really need to be testing those using ex vivo, in vitro systems. And I know that several of our mycology colleagues have been funded to do studies like that, especially now that Candida auris can be funded with the extra antibiotic resistance, NIH funds. So I don't have the answer to that, but I hope that when you invite back some of the mycology colleagues, some of John's colleagues actually in Duke are even working on that. Because I think those are those are definitely testable hypotheses. There's also really some beautiful work. So I know I did put that forward for another mycology talk from Lindsay Callan, who's been working on those kinds, looking at bacteria that can inhibit Candida auris. And I think we will find targets. That's one of the really fascinating parts of the human microbiome, is thinking about what are the molecules that commensals are producing in order to inhibit the colonization of more pathogenic organisms. So stay tuned for that. I think that what we're doing is trying to generate hypotheses, and some of them we're trying to test ourselves, but it's a big world, and we're happy if other people get there first, because there's a lot of ground to cover. And in terms of diabetes, absolutely correct that diabetes does change the skin microbial community. And you're just having a lot more glucose and sugar availability. And that's been really the work of a former postdoc in the lab who's really wonderful brilliant colleague at University of Pennsylvania, Elizabeth Grice, has been looking at looking at the bacteria and the fungi associated with diabetes.
Right, that'll be really, really interesting to follow. So at this point, the time is, we have 1 minute left, and so we've run out of time essentially. We will send all of the questions that haven't been, all of the questions, including those that were asked, but also those that we may not have reached, to the speakers so that they can actually connect with those who asked them if they want to. I'd like to thank the speakers, to Jay Knowles and to Julie and to John for making the time to do this today. And also to the MRC CMM staff, who are amazing at keeping the Mycotalks running smoothly and while staying very much in the background. We all look forward to seeing you on the 27th of April, when our speakers will be Yenping Su from Taiwan and Jun Quan Chung from the National Institute of Allergy and Infectious Disease in the US. So until then, take care and goodbye, good morning, good night, good afternoon. Bye-bye.